What's up, guys? Today I'm going to be going through 5 multiple choice questions that I made for a first year medicine mock. Okay, so question 1, plasma has an intrinsic pressure generated by plasma proteins. This acts to hold water within the capillaries. What is the name of this pressure? So A, hydrostatic, B, albuminary, C, hematinic, D, efferent flow, or E, colloid. So of course the answer is E, colloid pressure. Colloid pressure is mostly generated by this protein here, albumin. Very interesting protein, very important protein. And the reason it's so good at this job in particular is because it is very negatively charged. And if you look here, we see the polar water molecule, and the hydrogen atoms are more positively charged. So it's sort of like a directional charge, meaning the very negatively charged albumin is able to attract that water and sort of hold fluid within the plasma as blood goes through capillaries. And colloid pressure is a really important force that counteracts hydrostatic force caused by like the water pressure from your vascular system. So question 2, aldosterone. Which of these options does aldosterone release not lead to in the kidneys? So we have reabsorption of sodium, potassium chloride, water, or aldosterone promotes all the above. So the answer is potassium reabsorption. So let's think, why is aldosterone released in the first place? So the first reason is hypoperfusion of the kidneys caused by low blood pressure or blood loss, water loss. The kidneys respond by triggering the renin, angiotensin, aldosterone system. Aldosterone is released, and helpfully in that scenario, it triggers sodium reabsorption, chloride reabsorption, and water reabsorption to sort of try and restore blood volume. And then the other main reason why aldosterone can be released is when there's high levels of potassium in the blood, hyperkalemia. This can directly trigger the release of aldosterone from the adrenal cortex, and that aldosterone release acts to rid the body of that excess potassium. So we can say that, of course, it's not B because aldosterone is important for excreting potassium. Okay, guys, question 3. MastCellMulvaney is a medical student and is confused about what type of transporter SGLT1 or 2 is. Which of these is the best description? So A, antiporter removes sodium ions from the cell and brings in glucose molecules in exchange. B, insulin-dependent glucose transporter uses ATP to bring glucose molecules in. C, an ATP is symporter, uses 1 molecule of ATP to bring 1 sodium ion and 1 glucose molecule into the cell. Or D, a symporter which uses the electrochemical gradient to bring 1 sodium ion and 1 glucose molecule into the cell. So I mean, this is a case of knowing it really. If you don't know it, this is not going to be easy. So the answer is D, it is a symporter which uses the electrochemical gradient to bring 1 sodium ion and 1 glucose molecule into the cell. So here's a diagram. So this is in the proximal convoluted tubule of the kidneys. So blood gets filtered, filtrate, leaves Bowman's capsule, comes into the PCT. It's got loads of sodium, loads of glucose. So it's a really smart mechanism that the body uses here. So let's focus here for a second. So this is the classic sodium potassium ATP is. It's pumping sodium back into the bloodstream, pumping potassium into these cells. And what that does is, because there's so little sodium in here, because it's being pumped out, there's a really strong concentration gradient forcing sodium this way. So by creating that gradient artificially using the sodium potassium pump here, the body is sort of able to get glucose a free ride in with sodium. So it allows sodium in down its concentration gradient and is able to bring glucose in for free, basically. And then glucose taken out into the bloodstream as well. Okay, question 4. What is the action of the enzyme enterokinase? So once again, if you don't know, that's fine. The answer is B, cleaves trypsinogen to its active form trypsin. So enterokinase found in the duodenum brush border. The rest of these are created in the acinar cells of the pancreas. So pancreas releases these proenzymes. They end up in the duodenum. Enterokinase cleaves trypsinogen to trypsin. Then trypsin activates the rest of these enzymes. So the bonus question is, why is it important that these pancreatic enzymes don't get activated prematurely? Well, it's because if that happened, the pancreas would end up digesting itself. These are powerful proteases. If they were activated inside the pancreas, trapped inside the pancreas, that would cause a lot of problems, and actually is responsible for some cases of pancreatitis, like in the case of cystic fibrosis. Fatty acids undergo a metabolic process to be used for energy. This process involves converting them into acetyl coenzyme A. What is this process known as? So we know that this is catabolic. We're breaking down large molecules, using them for energy. 5 options here. This is an important one to know. The answer is beta oxidation. So the way this goes, beta oxidation involves fatty acids, these long carbon chains being brought into the mitochondrial matrix by carnitine shuttle, and these long carbon chains are broken down into 2 carbon chunks, these acetyl coenzyme A's, and then those can be fit straight into the Krebs cycle, the citric acid cycle, and be used to generate ATP. So those are my 5 questions. Thank you for watching.